in September 2013, just after the height of the UME, 16 and 19% respectively of dolphins were found to be underweight. By December 2013, it appears that the percentage of underweight dolphins had returned to pre-UME levels. So the working hypothesis for the cause of this UME is that there has been prey loss or a prey shift due to seagrass loss in preceding years. However, this investigation is still ongoing, and the next steps include conducting morbillivirus surveillance on a subset of dolphins and pursuing additional testing based on histology, biotoxin, and stable isotope results. The investigative teams are also coordinating with the Florida Fish and Wildlife Conservation Commission and others to evaluate whether there are any changes in prey distribution or composition. Finally, the last UME that I'll be discussing today is the Northern Gulf of Mexico cetacean UME. It's been going on for more than 4 years and is still ongoing. This UME is the largest die-off of bottlenose dolphins in the Gulf and is unprecedented both in duration and scale. Elevated strandings in Louisiana, Mississippi, Alabama, and the Florida Panhandle began in February 2010, and it includes 1,150 cetacean strandings as of about a week and a half ago. 86% of the strandings are bottlenose dolphins, and 95% of the animals have stranded dead. A couple months after the start of the UME, there was a fire and explosion on board the drilling rig Deepwater Horizon. I'm sure we all know about that, which resulted in an oil spill of national significance that flowed until July 2010. In total, approximately 210 million gallons of oil were spilled into the Gulf of Mexico. This figure shows the cumulative surface oiling from Deepwater Horizon, here in the gray, and the area defined in the UME in the red box. The Northern Gulf UME overlaps with the Deepwater Horizon oil spill in both its response area and the natural resource damage assessment. So what have the stranding rates in the Northern Gulf looked like over the past 4 years? This chart shows monthly strandings in red from February 2010 through December 2013. The blue bars show the historical monthly average for the UME area. The error bars represent 2 times the standard deviation. So although the UME start date is February 2010, strandings really picked up in March. From February to April, there were high stranding rates across the UME region, but especially in Lake Pontchartrain, Louisiana, where 26 dolphins stranded. These Lake Pontchartrain dolphin strandings were notable, since bottlenose dolphins are not thought to be residents in the lake and were rare prior to 2010. However, a group of 30 to 40 bottlenose dolphins had been observed regularly in the lake since 2007 following Hurricane Katrina, and we had been monitoring them with periodic photo identification surveys. It was also a really cold winter in 2010, and temperatures in the lake were lower than average, and salinities were also particularly low. So on April 20th, the Deepwater Horizon oil spill began, and stranding rates remained high for the UME area throughout the year. Historically, there's a spring peak of strandings in the Northern Gulf area, and this was also true in 2011. However, the magnitude of the spring peak was high, and there was a high proportion of small dolphins, less than 115 centimeters in length, that stranded between January and April. Stranding rates remained elevated in the summer and fall and were higher than they had been in 2010. 2012 was not as high as 2011, and the summer stranding rates fell down close to average, which got everyone really excited, but then they began to climb back up in the fall. The spring of 2013 was high again, with April being the highest on record. And stranding rates in the summer were once again back close to average, where we thought our UME might be ending, but as in previous years, they started to creep back up again in the fall. And although not pictured on this graph, February 2014 was the second highest recorded in the UME, second only to 2011. This chart shows only the strandings of bottlenose dolphins that were less than 115 centimeters in length by month, beginning in 2010 and continuing through early 2014. I'll be referring to this size class of dolphins as perinates. The chart shows the historical pattern of most perinate strandings occurring between January and May of each year. You can see the pattern is similar in 2011, which is shown here in yellow, but the magnitude of the strandings was much, much higher, especially in February and March. Most of these perinate strandings were in Mississippi and Alabama. Because strandings were elevated a couple months before the start of the oil spill, the UME investigative team took the approach of investigating previous causes of Gulf of Mexico UMEs first. This included testing for morbillivirus and biotoxins. Because of the high proportion of perinates in 2011, we also wanted to investigate the bacterium Brucella. Brucella and its associated disease brucellosis are known to cause abortion. While we began the investigation this way, we worked closely with the Working Group for Marine Mammal Unusual Mortality Events, that big long name, and our investigative team to develop a comprehensive sampling protocol that would ensure we have collected enough samples to investigate all other causes of the UME, including the oil spill. We began with a large-scale cetacean UME, but because 86% of the strandings were bottlenose dolphins, we decided to focus our analyses there. That said, we have closely investigated and sampled all non-bottlenose dolphin strandings in the Gulf, including the Gulf outside the UME area, as appropriate based on the decomposition state of the stranded animal. We've tested a subset of dolphins for morbillivirus and biotoxins, but to date, results don't indicate that either of these were contributing factors to this event. Brucella testing completed to date showed that 41% of perinates so far are positive, and 13% of non-perinates are positive. Historically, we've not routinely tested for Brucella during unusual mortality events, although recent testing indicates that it may be more common in U.S. waters than we previously thought. We're currently working with experts to get a better understanding of Brucella, what background levels of infection were in the Gulf prior to the UME, and its role in the ongoing UME. Next, we looked at demographics and compared that to historical stranding data. We reviewed all previous Gulf of Mexico cetacean UMEs in comparison to this one. The highest number of strandings in a previous event was 344 animals during the 1990 die-off. This one is well over 1,100. The longest in duration prior to this one was 17 months, and we're now over 4 years. 55% of previous UMEs in the Gulf were related to biotoxin, and 27% to morbillivirus. This one doesn't appear to be due to either. 2 previous events in the Gulf have had a high proportion of perinate strandings, and both were in Texas in 2007 and 2008. Brucella was suspected in those events, but we were unable to confirm due to limitations in diagnostic testing at the time and poor carcass condition. The role of Brucella in this event is still under investigation. And lastly, there's been no evidence of oil exposure in any previous event, but the role of the Deepwater Horizon oil spill in this event is still under investigation. Through modeling of this event, 3 primary clusters of elevated strandings were identified. These were Lake Pontchartrain, Louisiana in 2010 and 2013, Mississippi and Alabama in 2011, particularly those very high perinate strandings I showed you, and Louisiana throughout the entire event and for all age classes. Our next steps were to investigate each cluster using histopathology. For this, we focused on the freshest carcasses. So far, we've completed histopathology from 2010 and 2011, so I'm only going to be presenting those results. For non-perinates, we use histopathology available from reference populations to set up a case control study and evaluate which pathologies were unexpected. Samples for histology weren't available from Lake Pontchartrain in 2010 due to the high level of decomposition for those stranded animals. And cases available for 2013 haven't been analyzed yet, so we need to still investigate that cluster a little bit farther. In looking at the perinates from Mississippi and Alabama, histology indicated that many from this group either died in utero or did not completely inflate their lungs after birth. Many had in utero fetal distress, with or without Brucella-associated pneumonias, and multiple strains of marine Brucella were present in the affected animals. For the Louisiana non-perinate dolphins, a number had histologic evidence of bacterial pneumonia, and some of these cases were the most severe pneumonias that our pathologist had ever seen in dolphins. Dolphins in this Louisiana group also were statistically more likely to have bacterial pneumonia compared to control populations. And animals also had increased incidence of adrenal gland abnormalities compared to control populations. So where are we now in this investigation? To date, we've shown that this UME is unprecedented, both in magnitude and duration, compared to previous UMEs in the Gulf, and the most common factors of previous UMEs, a brevetoxin and morbillivirus, don't appear to be contributing to this event. In the non-perinates in 2010 and 2011, which were mostly from Louisiana, we found an increased risk of bacterial pneumonia and adrenal gland abnormalities. Secondly, in the perinates in 2010 and 2011, we saw a high incidence of fetal distress and pneumonia, which in some cases was caused by Brucella infection. The cause of this event remains unknown, and the role of the Deepwater Horizon oil spill in causing or amplifying the severity and duration of this UME is still under investigation. However, it's worth noting that simultaneous to our UME investigation, as part of the Deepwater Horizon oil spill natural resource damage assessment, there have been studies on bottlenose dolphins in the northern Gulf. In particular, there have been bottlenose dolphin health assessments conducted for free-swimming dolphins in Barataria Bay, Louisiana, which received heavy and prolonged oiling during the Deepwater Horizon spill, which is located kind of right up in there, and also in Sarasota Bay, Florida, as a control site that did not receive oiling. During these health assessments, dolphins were temporarily captured, given veterinary examinations, and released. And the results from this study were recently published in the Journal of Environmental Science and Technology, and I'll briefly review them here. So Barataria Bay dolphins demonstrated a number of relatively uncommon disease conditions consistent with effects noted previously in marine wildlife following oil spills and in experimental studies of petroleum hydrocarbon toxicity in mammals. These included decreased concentrations of serum stress hormones called hypoadrenocorticism. They were 5 times more likely to have moderate to severe lung disease. There was a high prevalence of blood abnormalities, including hypoglycemia, anemia, and inflammation blood parameters. The animals were in overall poor body condition, with 25% of animals being underweight. Some animals exhibited nearly complete or complete tooth loss, and 48% of the dolphins sampled were given a guarded or worse prognosis, and 17% were graded as poor or grave, meaning they were not expected to survive. From this study, a conceptual model was developed for health effects previously reported for mammals in association with oil exposure, and those are shown here in the yellow boxes. They include adrenal effects, liver injury, lung injury, and maternal transfer to young. And these effects could lead to the abnormalities observed in the Barataria Bay dolphins in the blue boxes, and ultimately to endpoints, such as decreased survival, increased disease susceptibility, and fetal death. So, for example, if dolphins were exposed to oil, you would expect to see adrenal effects, which were in fact measured in the form of decreased mineralocorticoids or decreased serum aldosterone and decreased glucocorticoids or decreased serum cortisol during a stress response. Many of the abnormalities and endpoints observed in the Barataria Bay dolphins are similar to those observed from stranded dolphins in our unusual mortality event. So where are we now? Let's revisit this map of our UMEs. We found that the Mid-Atlantic UME is clearly due to an outbreak of morbillivirus. The cause of the Indian River Lagoon bottlenose dolphin UME is still under investigation, but it may be due to ecological factors such as prey depletion or shifts as a result of massive seagrass die-offs. And the cause of the Northern Gulf UME, or the causes, are still under investigation, but it may be related to or exacerbated by the Deepwater Horizon oil spill. So why do all these dolphin deaths matter? Aside from the fact that we all really like bottlenose dolphins and we like to see them when we're out on the water or at the beach, dolphins and other marine mammals are considered sentinel species. They're similar to the canary in a coal mine. What makes them a prime sentinel species? Well, they're long-lived, like us. They feed at a high trophic level, like us. They reside in a coastal environment next to which 39% of the U.S. population resides, and they have unique fat stores, which serve as a depot for anthropogenic toxins. So we might not have blubber, but we certainly are susceptible to fat-soluble contaminants. What does it mean to be a sentinel species? Marine mammals can tell us about ecosystem health and changing environmental conditions that could ultimately impact humans in a number of ways, but I'll review a few. Shifting migratory patterns can tell us about changing ocean temperatures and underlying ecosystems. For example, there's evidence that gray whales in the Pacific might be delaying their southbound migration and even remaining in polar waters over winter. Changes in marine mammal body condition can tell us about shifts in the prey base, food web, or ecosystem. For example, the poster child for these changes is the declining body size and condition of some Arctic polar bears due to declining sea ice. Emerging infectious diseases or alteration in pathogen transmission can indicate new sources of risk for animal and human health. For example, endangered sea otters in California have been found to suffer lethal infections from a parasite, Toxoplasma gondii, that's shed in cat feces. Their illness illustrates the potential for terrestrial diseases to spread to the marine environment via polluted runoff. Finally, marine mammals exposed to toxicants provide information on the chemicals that present the greatest risk to consumers at the top of the food chain. For example, 18% of stranded dead adult California sea lions have been found to have urogenital cancer. This very high prevalence of cancer has been attributed to high concentrations of organochlorines, or PCBs, in their blubber in combination with other factors. So let's go back to our unusual mortality events just one last time. Are they ocean health indicators from a sentinel species, or are they just unusual events? At this point, it might be too early to say whether or not the UMEs in 2013 illuminate larger ocean health issues. However, it's clear that bottlenose dolphins are currently facing a number of challenges and pressures in their marine environment, and populations will be recovering for many years to come. Thanks so much.
Where you see the severe bacterial pneumonia?
Yeah.
Have you guys looked at which types of bacteria it is, or does it seem like it's one pathogen, or does it seem more like it's an opportunistic infection?
Yeah, they have done some looking at that, and I know that that a very, very small number are due to brucella, so we don't typically see. So in the perinates, a lot of it is due to brucella. In the non-perinates, I think it's only like 4 or 5 or 6 animals that have had brucella, so it's a very low proportion of the older animals that are showing that. It is bacterial pneumonias, and I'm just not sure which specific bacteria. I can find out for you, but I think it's multiple strains.
Can I ask one more question?
Mm-hmm.
So with the morbillivirus, is vaccinating dolphins a reasonable, feasible, or ridiculous strategy?
It's something that people talk about a lot, but there's just no effective or kind of logistical. First of all, we don't have a vaccination for morbillivirus yet. It's something that a lot of people who work with captive dolphins are really interested in having because it is so highly lethal when animals are exposed to it. So it's something the Navy is really interested in for their dolphins, obviously, and something that captive dolphin people would like to see. But currently there is no morbillivirus vaccine. So even if there was one, having a delivery mechanism to get it out to thousands and thousands of free-swimming dolphins is something that would be highly complicated. So I would say we're far from that now. But that is, if you could vaccinate them, yes, that would protect them because the animals that are left obviously have developed an immunity. Not every animal dies. It's kind of like why we get vaccinated, so you increase your herd immunity. But yeah.
Have you found elevated levels of PAH in any of your dolphins in Mexico?
So I don't have those results for me. We have looked at that, but I don't have those results available right now. From what I understand, I think that it can be complicated depending on where you're getting those samples from. So if you're getting it from bile or from different tissues, and I think it degrades at different rates in the body. So but that's not my area of expertise. I know we've looked at that, but I don't have that information available. Yeah.
Explain one more time for the North Gulf UMB why you see the sweeping increases in strandings during the winter months and early spring.
We don't know. Typically, we do get a spring peak of strandings, and normally that's because it's the calving season, and so there's a lot of mortality associated with the calving season, calves that don't make it, or moms that are dying during childbirth. So we do typically get kind of a spring peak in strandings, but never as high as we've seen in the Northern Gulf. over the past 4 years. And also the event starting, as you noted, earlier in the year. So typically it's kind of March, April. Those really, really high strandings are starting earlier in the year in January. Some of that may be due to the aborted fetuses that we're seeing or the stillborn animals that are being born. But it's not all attributable to perinates. So it's a mixture, and we just don't know why.
The morbidity, which is saying the detection, you got 4 different tests that you do. Now, I'm assuming due to cost, the antibodies test, that's the first test, doesn't pass that. You don't waste the resources to continue testing?
So, yes, so for that, we actually, to have a confirmed case of morbillivirus, it has to pass a certain number of tests. So you're right, we do the first screening for the antibody. If it doesn't have the antibody, no need to go any further. But the antibody just tells you that it's been exposed at some point in the past, not that it has a current infection or that, that the disease caused death. So what we have to have in order to confirm morbillivirus, so when we have those suspect or confirmed cases, to confirm morbillivirus, you have to have either immunohistochemistry or PCR and lesions that are compatible with morbillivirus clinical disease upon histopathology. So for animals, for some of those other species that we tested where we don't know, they weren't actually ever moved to the confirmed category, some of that may be due to decomposition. We just don't have good histopathology for some of those animals. So we're never actually able to say confirmed morbillivirus. So yes, you need to have either PCR and immunohistochemistry and the histopathology confirmed to be able to call it confirmed morbillivirus. And you're right, it's really expensive, which is another reason we don't test every stranding for it. So.
Is there any good news with the rehabilitation of the animals that are taken in?
We are not taking in any animals for rehab from the morbillivirus event. So because morbillivirus is so highly contagious and lethal, a lot of our facilities that do rehab also have captive animals in their collections. And so we've forbidden any organization to rehab a morbillivirus, an animal that's stranded in the morbillivirus area. Also, because we don't really know very much about how morbillivirus acts in the body itself, it's really considered, we don't know if we can rehab them. So it's more research versus stranding response and rehabilitation. So it's really more of a research project. And there are organizations, like I said, the Navy would be very interested in knowing if you can rehab a dolphin. But we don't know how long animals shed the virus. We don't know which tissues shed the virus. So even if we rehabbed an animal, it could theoretically still be shedding virus, and you don't want to put it back then out into the environment where it could infect other dolphins. So it's just not something we've been doing. In other areas, we did rehab a dolphin from the Indian River Lagoon that live stranded during that event, and it's been released and it's back out in the wild doing really well. It was rehabbed at SeaWorld in Orlando and was re-released to the lagoon maybe back in December or something, I think. But it's doing well. It's been resighted. For the Northern Gulf, most of the animals have stranded